Welcome back to the Felix Factor with me, Felix Aaron. This week we're going to do a 3 triple section video. First of all, cell transport, then energy processing using cellular respiration and photosynthesis. It's pretty exciting. Diffusion is the movement of a substance down its concentration gradient. Here we see a membrane that is permeable to those little round circle things. As you can see, there are a lot of little round circle things on the left side, and fewer on the right side. What this means is that since the little round circle things, which in this case represent little molecules, are constantly kicking around, they will end up being a net movement down the concentration gradient to the right side. What this means is that if there are more things on one side than the other side, then there's a concentration gradient. And since there are more, you get more kicking and you get a net movement until it reaches equilibrium when there are just the same amount of little particles on both sides. Passive transport is when things move across a membrane that by diffusion, that is, they don't need any work. For example, oxygen diffuses out of your capillaries and into cells that use it to create or to metabolize ATP. Osmosis is simply a diffusion of water across a concentration gradient or across a semi-permeable membrane. Here we see another beautifully drawn semi-permeable membrane. On the right side we see many water molecules, but most of them are bound to by little green molecules, little green sugars. That makes the water molecules unpermeable to the semi-permeable membrane. On the left side, however, we see water molecules which are permeable, and even though there are more molecules on the right side, those cannot pass through because they are bound to. And so, even though there are more water molecules on the right side, there are more free molecules on the left side, so there is a concentration gradient, and therefore there is a net movement from left to right. Tonicity is usually used when describing a solution's ability to have a cell gain or lose water to its environment. If I've been exercising, that means my body is letting go lots of salts via sweat. Therefore, my cells have less water in them. That makes a concentration gradient, and since there is more water inside of my cell than outside, that means the water in my cells will go out to balance it. This is what we call a hypertonic solution, which means there are more solutes outside of the cell than in the cell, which causes water, of course, to then leave the cell and go out. However, if I'm in a pickle eating contest, I'm eating a lot of pickles, that will cause an increase in salts and other solutes in my cells, which will mean that a lot of water will go into my cells to balance out the concentration gradient. This is called a hypotonic solution, when there are fewer solutes outside the cell as inside. Seen in another light, eating a lot of pickles will cause an influx of salt into my bloodstream, which will make all the water in my cell go out. However, in a plant, when there's a lot of water in the cell, that makes it turgid, which just makes it nice and strong, which is good for the plant cell. When there is water, not enough water, then it gets shriveled. All this taking in and taking out of water is all called osmoregulation. Facilitated diffusion is when things that cannot ordinarily pass through cells very easily, for example, polar molecules that can't go through the hydrophobic middle of the phospholipid bilayer, can go through other things that are little tubes through which they can pass. This is called facilitated diffusion. An example of this is water, which, being polar, has trouble going through the hydrophobic middle of the phospholipid bilayer. Thus, it goes through aquaporins, which are little tubes through which water can pass and diffuse a lot faster, since cells need to diffuse lots of water in and out. These aquaporins make facilitated diffusion a lot more facile. Remember last time you skinned your knee? Remember how it hurt a lot? Well, that was all because of your nervous system transmitting that signal to your brain so it can tell you that your knee was skinned and it hurt. Now, you might wonder how this connects, and the answer is that the nervous system is run using nerve impulses, and that has to happen because of things called uh, sodium potassium pumps. These things pump sodium potassium across the cell membrane, and if it when it gets an impulse, and if it pumps enough, that reaches a certain level, which sends a signal to your brain. These sodium potassium pumps, however, use active transport, which means that there's a lot of them on one side, and when it pushes them to the other side against its concentration gradient, that is what helps to transmit a nerve signal. This is only possible with facilitated diffusion, which takes ATP, which takes energy, because that is pushing things against their concentration gradient. The two main pillars of cell transport are these: exocytosis and endocytosis. Plainly put, exocytosis is kicking stuff out. Endocytosis is bringing stuff in. However, endocytosis gets a little more specific. First, we have phagocytosis, which is cellular eating. For example, cells eat glucose that's floating around in your bloodstream. They take it in. They put it in vesicles, and then it's in. Penocytosis is cellular drinking. Non-specifically, it takes in liquid, puts it in vesicles, and then it's in. However, as with phagocytosis, when things get digested in the lysosomes, that doesn't necessarily have to happen in penocytosis. Receptor-mediated endocytosis is when cells have receptors on their membranes, and they use that to take in substances, for example, lipids from the blood, to synthesize proteins with. When two substrates want to react, it often takes a lot of energy to do so. So what enzymes basically do is they fit the two substrates together so that the energy of activation is a lot lower. This makes reactions go by a lot faster, and you need to use a lot less energy to catalyze them. So let's just say this molecule and this molecule want to react. What happens is you have the active site, which is where they happen, and then they stick in there and they react, which I must say happens using the induced fit, which kind of squeezes them into shape to make them work. Also, we have things called cofactors and coenzymes. These are little things that help the enzyme out when catalyzing reactions. If they are non-organic, then they are called cofactors. If they are organic, they're called coenzymes. And this might seem all good and peachy to you, but there's a couple of things you have to remember. If enzymes get too hot, they'll get denatured, because the jiggling of their molecules will and their three-dimensional shape, well, needless to say, it will become all wrinkled and stuff, and now, when these two substrates had to fit in, doesn't work. Also, we have a high pH or low pH, difference in pH, and a high salinity levels, because what this does is it creates a lot of ions, and that makes that weakens the hydrogen bonds and makes the enzymes no longer catalyze well. Now, you may have heard that cellular respiration is for animals and photosynthesis is for plants. Well, if you heard that, then you're partially right. However, you're not all the way right. If you want to be all the way right, then learn this. Cellular respiration is in plants too, because all plants need ATP, which is one of the biggest outcomes, or one of the biggest products of cellular respiration. Now, since respiration and cellular respiration sound so similar, you might be tempted to believe that they are the same thing. But do not think so, because they are not. Respiration is taking oxygen into your lungs and then distributing that throughout your bloodstream to your cells. Cellular respiration is using that oxygen to then create ATP. You see, the equation for cellular respiration looks like this: 6O2 plus C6H12O6, which is glucose, or other forms of carbohydrates, reacts to yield 6CO2 plus 6H2O plus ATP. Photosynthesis is the opposite. it takes carbon dioxide and water and produces with that carbohydrates like glucose and oxygen. Alright, so this one's a big bopper, so fasten your seatbelts and get ready. Cellular respiration starts out with a process called glycolysis, which takes a 6-carbon molecule, glucose, and splits it in half, forming 2 3-carbon pyruvates. This takes 2 ATPs to do, but it generates 4, so that's a net plus 2 ATPs. Next, your 3-carbon molecule. You watch as one of the carbons gets taken off, and a water molecule, pretty small, gets added onto there. That makes acetyl-CoA, which is the product that will go into the Krebs cycle. Now, acetyl-CoA is ready to enter the Krebs cycle. What happens first is the water molecule gets lopped off, attached to a 4-carbon molecule right here. Here is our new 6-carbon chain thing that is not really sticking together. Come on, thing. Come on. Okay, good. Here's a 6-carbon chain that in real life is probably a lot smoother than it is right now. So what happens first is 2 carbon dioxides are generated, but 3 NADHs during this product, NAD pluses, oxidized, 2 electrons plus a hydrogen proton lopped on there. You get NADH. You get 3 of those during this cycle. And those are very helpful. Those will be used during oxidative phosphorylation in the next step. And also, 1 ATP is produced. Also, 1 FAD is plus 1 hydrogen plus 2 electrons. That is oxidized on there. Then you get 1 FADH2. So net, we get 3 NADHs, 1 FADH2, and 1 ATP. Then you get the last 2 carbons lopped off there. And here we got a 4-carbon chain to be used again with acetyl-CoA again. Isn't that fantastic? I forgot to mention that after the carbon dioxide gets taken off, that makes the ion negatively charged, which is where the electrons get funded for the oxidation of the NADHs, the 3 of those, and the 1 FADH dose. Finally, we have reached oxidative phosphorylation. This is where the real ATP is generated. So here we have an electron that gets taken off of an NADH, an NADH. It reduces it, electron comes off, attaches onto this higher affinity protein. Then, once it does that, the protein takes a bit of energy off of it and pumps a hydrogen proton, out of the matrix into the outside of the cell, of the mitochondria, I mean. Then, gets passed along to more affinity proteins. Electrons keep being used for proton pumping until finally it gets to this last one where all the energy is taken out. Then the electron goes to an H2O, and it uses that to bond to the hydrogen. That creates a hydrogen, and that's why O2 is used in this reaction. That's why O2 is needed. Now, since we have all the hydrogens in the intermembrane spot, you get a hydrogen transport thing, like a ATP synthase, which then, since there's a hydrogen gradient, you get the gradient moving, so you get the protons that go inside. They go inside, and that spins a little spin thing. It goes around and around and around. Then you get ADP, which gets smashed together with a single single phosphate group, and that creates ATP. And there is how ATP is generated. 38 ideally, but really fewer than that usually, because cells aren't 100% efficient. Now in this world, there are 3 main types of animals. First we have autotrophs. These autotrophs are beings that create their own food from sunlight. For example, we have plants. Those are autotrophs. They make their own energy from sunlight. Or we have photosynthetic bacteria. Or we have chemoautotrophic zooplankton that live next to the hot vapor things in the bottom of the ocean. They make their energy for themselves. Those are what autotrophs are. Also, another word for these is producers, because of course they produce their own food. And another even smaller way of funneling these things in is called photoautotrophs. And those are beings like Protista and Plantae that make their energy strictly out of sunlight and photons. So this is a leaf, or at least it's supposed to be. And at the top you see the palisade mesophyll, at the bottom the spongy mesophyll. And then you'll notice that at the certain parts of the outside of the leaf there are little things called stomata, through which gases get exchanged like oxygen as waste product and carbon dioxide as fuel. Vascular bundles supply water to the chloroplasts, which are then in turn located in the palisade mesophyll. This is a chloroplast. As you can see, chloroplasts are made of little stacks of thylakoids called grana. Each granum consists of several thylakoids, which you can see there. In each side of each thylakoid is something called the thylakoid space, and outside of each granum is something called the stroma. This will all make more sense later. What is important to note is that in the thylakoids, there are little embedded proteins called chlorophyll. And these are the things that actually capture the sunlight, and that is where the ATP is made for the dark reactions, which then happen in the stroma. When Felix takes his Cubs hat off, you know it's serious. Serious because we're discussing what happens during photosynthesis. So as we know, the general equation for photosynthesis is 6 carbon dioxides, 12 waters, react to yield 1 glucose, 6 waters, and 6 O2. If you want to do net, then you really don't have to have the 6 H2Os on the other side, but technically water is created, even though it is less than is used. So that's a thing. But as you'll notice, in the beginning of photosynthesis, we have the H2O gets lysed, the hydrogens, protons go flinging out, and the oxygens get absorbed or get flown away as O2 gas as a byproduct, and the NADP pluses get reduced to become NADPH, which become then electron carriers. So it's important to remember that these are the oxygens that get lysed off the water, that those become the byproduct oxygen, which is why when scientists use O18 oxygen in their water molecules, that's when they notice that they get O18 coming out as byproduct, whereas if you use the O18 in your CO2, then you won't get that because the CO2 gets used to form the glucose, and the O2, the O part at least, that part, and then the other O gets put into the water molecule at the end of the Calvin cycle. Alright, enough of that. Let's get this going. So, we start off with a chlorophyll molecule. Just chilling, not doing much. Happens upon a photon. The photon comes whoop, screaming in. It energizes one of these electrons on here. It gets really energized and it goes. Then, that electron jumps from this one to another energy carrier which has a higher affinity. This energy carrier, by luck, happens to be an energy carrier that uses the energy from this electron. It pumps a proton out into the thylakoid membrane. Then happens upon another one. Oh, another one. Same thing happens. Finally, it gets to photosystem uno, where another photon hits it, gets really excited again, and then it happens upon an NAD plus P plus. Now, this NADP plus is in need of an H plus and an electron to reduce it. Well, guess what happens? There's a water molecule that happens back across the chlorophyll in photosystem 2. And the photosystem 2 is saying, oh darn, I need my electron back, it left. And water is saying, okay, I'll give you one. And so the water, in a very romantic and heartfelt tribute, takes its two oxygens off. Its two hydrogens off its oxygen. The oxygen pairs with another oxygen. It goes away. And the hydrogens, well, they give up their electrons. One of them goes on to here, on back to the chlorophyll, so the chlorophyll is happy again. One of them goes to the NADP plus molecule, which needs to get oxidized using that other electron on photosystem 1 and the other hydrogen. So those pop on there. You get a happy NADPH, a happy photosystem 2, a happy photosystem 1, and happy hydrogen atoms and happiness everywhere. Yay! So those are the light reactions. Now in the dark reaction, this is where it gets a little tricky. So we start off with our 5-carbon strand. This is the thing that starts off the dark Kelvin cycle. So we take this, we get a carbon atom from a carbon dioxide that happens to be passing by. It sticks on there. You see how it's sticking on there? Yes, you do, because it is. And that becomes now a 6-carbon chain. Now what happens to this 6-carbon chain is it's very unstable. So it becomes 2 3-carbon chains. Now the 3-carbon chains, those, get moved around until they become a phosphoglyceraldehydes, which is basically half of glucoses. They connect together, and what do you know? We have a starch, or a glucose molecule, that can be used for the plant. Now, if you remember those oxygens that got taken off the CO2, well, those each find some happy hydrogens. They bind together, and voila, we get water. That's photosynthesis for you. So we all know that chlorophyll is a molecule that gets the photon, kind of like the really popular guys who always get, the girls. This chlorophyll always gets the photons. Kind of a funky analogy, but whatever. That's the way it is. So these chlorophyll, which seem green, actually their deal is that they take in all types of wavelengths of light, like for example blue, violet, and red light, but it reflects the green light, and that is why we think they're green. It's how pigment works, which is pretty cool. There are actually 2 types of these: chlorophyll A and chlorophyll B, and both of these take in different wavelengths of light to be used in electron excitation. However, there is another type of pigment thing. It's called a carotenoid. It can also take in other things, but one of its more important jobs is to protect us from excess light. It can expel different wavelengths, which is one of the reasons why leaves turn orange in the fall. Carotenoid, kind of like carrot, orange. Aha! That's cool. So in case you haven't had enough of me in bio this year, we're going to do one more great recap section, photosynthesis. So starting it off again, we have our photosystem 2. It has chlorophyll. The chlorophyll donates an electron very graciously, which gets excited by a photon, climbs onto a high affinity proton thing, pumps a proton into the membrane, keeps doing that till it loses energy, climbs on a photosystem 1. Then guess what happens to photosystem 1? I'll bet you can guess. Gets excited by another photon that climbs onto an NADP+, gets reduced by the electron from there, from water, which gets split and returns one electron to the photosystem too, and also the H plus gets then donated to the NADP plus, becomes NADPH, goes into the Calvin cycle. Calvin cycle makes 6 carbon chains, reduces them to phosphoglyceraldehyde. Phosphoglyceraldehydes can then become either starches for usage to make food or cellulose to strengthen your cells and stuff. All good stuff. Remember, this is the most important thing for our lives in terms of our food and our daily happiness, because without this stuff, think about how long you would last without breathing in oxygen. I will try it now. You can watch. You see, I couldn't even last a minute. Without plants photosynthesizing and creating oxygen, I couldn't breathe. Plus, what would I eat? Would I eat dirt? That's kind of gross. Would I eat like rocks? No, rocks aren't tasty. I mean, I could eat a lot of water, but that's not very filling. So, you can see, without photosynthesis, we would not exist. Our favorite fish, he couldn't exist, right? Photosynthesis is the most important chemical reaction in the world. And so I leave you on that note. Have a good year, and I will see you in chemistry.